Hi, in this presentation I will discuss how to improve the variational phylogenetic parameters inference by learning the prior densities. My name is Amine Remita, PhD student at University du Québec à Montréal. This work was done under the supervision of Professor Abdoulaye Bani Diallo and with the help of my colleague Vite. The goal of the phylogenetic inference is to predict parameters that can explain the evolutionary history of a set of related biological sequences like DNA, RNA, and proteins. We can formulate the problem of phylogenetic parameters inference as follows. Given a multiple sequence alignment and a tree topology, which are the observable data, we want to infer a set of parameters omega, including the vector of the branch lengths and the parameters of a substitution model like KAT and GTR models. This brings us to model the conditional probability of the parameters omega given the data and the tree. Here we are interested in Bayesian inference. In this setting, the conditional probability of the parameters omega given the data and the tree can be computed using the Bayes rule. This probability is called the posterior and is given by the product of the likelihood of the data given the parameters and the tree and the probability of these parameters divided by the evidence, which is the probability of the data given the tree. First, the likelihood is evaluated using Fisher-Stein's pruning algorithm, which is a post-order traversal algorithm. Second, the joint prior density is represented by independent and fixed distributions over the parameters. Usually, in Bayesian phylogenetic software, we use default hyperparameters for prior distributions, such as the exponential distribution with a rate of 10 for each branch length of the tree. Finally, the evidence, also called the marginal likelihood, is calculated by marginalizing the joint probability of the data and the parameters over the values of all the parameters. Typically, multiple evolutionary parameters are jointly represented with different types of high-dimensional distributions, which induces computationally intractable evidence, which it makes difficult to compute the posterior. So Bayesian inference provides methods to efficiently approximate the intractable posterior and quantify the uncertainty in estimating the parameters. Among these methods, we find the Markov chain Monte Carlo, state-of-the-art in approximating the posterior. Another alternative is the variational inference, which started to gain some attraction from the phylogenetic community. Variational inference relies on fast optimization. First, it uses a new distribution Q called approximate distribution, which is chosen to be tractable from a known distribution family like normal, gamma, or divisional distributions. Second, it finds the optimal candidate from the space of Q that minimizes the Kullback-Leibler divergence to the exact posterior. However, we still need to compute the posterior, which is intractable. To overcome this problem, VI estimates a lower bound on the evidence called the elbow. If we develop the elbow, we find that it is composed of the expectation of the log likelihood, the data, and the divergence between the approximate density Q and the joint prior density of the parameters, which acts as a regularizer in maximizing the log likelihood. And with this new equation, we can avoid computing the posterior directly. So in order to minimize the divergence between the approximate and the true posterior, we only need to maximize the elbow with respect to the approximate parameters, using only the log likelihood and the prior density of the parameters. Since the likelihood is not in closed form, which is computed using a post-order traversal algorithm, we can evaluate its expectation by sampling from the variational distribution Q. Finally, we update the variational parameters of Q using stochastic gradient asset. One of the challenges we can encounter in Bayesian inference is the definition of an appropriate prior density for each parameter. It has been shown that using fixed prior densities biases the approximation of the true posterior and induces high probabilities. It gets worse when the data sets are small, the sequences are similar, corresponding to small branch lengths, or when we have correlated parameters. For example, in this figure, shows the prediction of branch lengths using 3 variational phylogenetic models with fixed exponential priors over the branches, each model with different rates. The dashed line corresponds to the true branch length mean of the sequences, which approximately corresponds to 0.001. We can notice that the VP models predict 2 times the true branch lengths, even when using the true rate as prior hyperparameter. So our objective in this study is to find a way to improve the variational posterior approximation by making the prior density more flexible and leveraging the structure and the optimization of the algorithm. This is a graphical illustration of a GTR-based variational phylogenetic model with fixed prior densities over the parameters. The parameters to be inferred are represented by white circles, the branch length, the substitution rates, and the relative frequencies. Each variational distribution is represented by a normal distribution, and its parameters, mu and sigma, are updated using stochastic gradient ascent during the inference. The gray squares are the predefined hyperparameters of the prior densities over the parameters which are fixed. This is the baseline model, and I call it the VBFPD model. A simple way to relax the rigidity of the prior densities is to define adaptable parameters of the density of the prior densities. These new parameters will be updated jointly with the variational parameters using stochastic gradient ascent during the inference. Starting the optimization, each new prior parameter is initialized using using an uniform sampled values. I will call this model the VPLPD model. To add more flexibility to the prior densities, we thought of using feed-forward neural networks to generate the values of these parameters from a uniform sampled input zetas. The weights and the biases of each neural network will be updated jointly with the variational parameters during the inference using gradient ascent. This is the VPNPD model. We evaluated and compared the proposed variational models in estimating 3 phylogenetic parameters: branch lengths with the Jukes counter model and substitution rates and relative frequencies of a GTR model. We simulated multiple sequence alignments using different lengths and sizes, and we replicated each alignment 100 times and fitted each alignment 10 times. We implemented and employed the NN3VB framework to perform the simulations and the inference. Finally, I will report the evaluation results in terms of the normalized Euclidean distance and the coefficient correlation between the real values used to simulate the data and the estimated values of the parameters. If we go back to the example I've shown previously and run the LPD and the NPD models, the new models with flexible prior densities, we can clearly see that the estimation of the branch lengths using the models with adaptable priors is closer to the true value than those estimated using models with fixed prior densities. Also, the variance of the estimations with the LPD and the NPD models is smaller. For more detailed results, we simulated 3 data sets with different branch length means: 0.001, generating highly similar sequences, 0.01, and finally 0.1, which produces divergent sequences. We infer the branch lengths using the 3 variational models. The FPD model with fixed prior densities uses fixed prior densities over the branch lengths with a mean of 0.1 for the 3 data sets. We can notice when the true prior means are different from 0.1, so 0.01 and 0.01, the LPD and the NPD models estimate the branch lengths better. The distance of the estimations to the true values are smaller than the FPD results, and their correlations are better. In the case of the true mean is 0.1, the third case, the estimations of the FPD models are closer to the true values, but the NPD and the NPD models estimated better correlated values. For the estimation of substitution rates of the GTR model, we simulated 3 data sets with different mutation rates. Using the rate ratio kappa, we controlled the difference between the two types of mutations, transitions and transversions. We can notice, independently of the dataset case, the estimations made by the NPD and the NPD models are improved compared to those made by the FPD models. They are closer and better correlated with the true values. Finally, we can make the same observations in estimating the relative frequencies. The LPD and the NPD models have better estimations in terms of the distance and correlation with real values. This is true even when the datasets are simulated with different frequency distributions. In conclusion, I presented how to use variational inference for estimating phylogenetic parameters. I showed that the variational phylogenetic models with fixed prior densities could be biased in approximating the posterior of the parameters. To overcome this issue, I presented a new way to provide flexibility to the prior using two updating strategies. To go further, we want to capture the correlations between the parameters and assess different neural network architectures, and finally implement adaptable prior densities over the tree topologies. You can find more details about the models and the results in the paper at this link. I want to thank my colleagues at the Laboratoire de Bioinformatique at Université du Québec à Montréal for discussing the methods and their support. Also, the federal and provincial funding agencies and Calcul Québec for providing computing resources. Thank you.